Our lab majorly work on mucosal immunology, and today I would like to introduce the gut microbiota and the epigenetics in the modulation of a lymphocyte in homeostasis. Okay. And the mucosal immune system compromises the body's largest immune system in the mucosal site. The mucosal tissues, including the gastrointestinal tract and the respiratory tract and the urogenic tract, faces a lot of pathogens or energies in the environment. And when we face the pathogens, just like previous talk, in our book, the immune system will activate it. At first, the innate immunity will activate, and the DC cells will capture the pathogens, the antigens, and translocate it to the lymph node. And the T cells and B cells will be activated by the DC cells. The activated T and B cells will traffic back to the infected site and contribute to the elimination of the pathogens. This is a process of how the immune system works during the infection. However, a lot of commensal bacteria coexist in our gut, and they contribute not only against pathogens, but also contribute to the development of the immune system and the function and establish the function of the immune system. The dysfunction or the dysbiosis of the microbiota will lead to the autoimmune disease or the energy or the metabolic syndromes. So we may say that both the mucosal immune system will face the pathogens and also communicate with the commensal bacteria. So as the surface of the mucosal immune system will face a lot of environment signals, not also from the diet nutrient, also the commensal bacteria and also the pathogens. That's environment signals. And this signal will transduct to the immune cells and change the epigenetics or the signaling of the immune cells. Then the function of the immune cells will change. So, and they regulate the disease progress, such as the imbalance of the immune system will lead to the inflammation, or the inhibitory of the immune system will lead to the cancer or etc. So this is signaling from the outside to the inside pathway. Then the first part I want to introduce to you is about how the microbiota or the commensal bacteria affect the intra-signaling or the function of the immune system. So how do the commensal bacteria modulate the mucosal tissue homeostasis and the immune cell function? This is the first question. As I just mentioned, the commensal bacteria, the microbiota, will educate or contribute to the development of the immune system. As you may know, that the numbers of the commensal bacteria may be 10 times more than our human cells, so they coexist with our bodies for a long time. And the dysfunction or dysbiosis of the microbiota will lead to not only the infection, inflammation, and the cancer. Our group majorly want to know how this commensal bacteria affect the tumor growth. Maybe we may first see the tumor environment. The tumor environment not only contains the tumor cells, but also the blood vessel and the metabolites, also the immune cells. And the development of the tumor, just like the old saying, Rome was not built in one day. At an early stage, the transformed cells, which means the cell with some mutant appeared, and the immune system, such as the NK cell, the natural killer cell, and the CD8 T cells will recognize them and kill them or eliminate them and keep the balance of our immune system. But however, during the immune editing of the inflammatory environment, the immune cells will highly express some inhibitory receptors, or the tumor cells will escape the immune surveillance of this immune cell, so the cancer cannot. So the process of the tumor progress is a very long and very dynamic process. So we want to know what's the function of the microbiota in this process. That is, what's the composition of the microbiota in the tissues. If there are some microbiotas in the tissues, what's the function of this microbiota? To answer these questions, we're looking to the colon cancer patient, and we collect some tissue samples from this patient. We collect the normal tissues and paratumor tissues, and also the tumor tissues from the same patients. And we analyze the intra-tissue bacteria of this patient from the same person, so that we can exclude the individual differences of different person. And we may know that different person have different diet, habit, or ages. But if we compare the same tissues, we may know the real differences between these tissues. And we did the 16S sequence to analyze the intra-tissue bacteria in these tissues. And we found that the Lactobacillus family bacteria, such as the Dora, Blautier, and the Ruminococcus bacteria, are enriched in the normal tissues. For the community network analysis, we also find that the Ruminococcus Blautier, the products and the Blautier bacteria are in the composition of this community network. So that this means that this bacteria maybe have some functions in the normal tissues. Next, we want to know what's the function of this bacteria. We use the subcutaneous mouse, tumor mouse model, and we inject the bacteria into the tumors, and we found that in the injection of this bacteria can inhibit the progress of the tumor growth, both in the subcutaneous tumor mouse model and also the spontaneous mouse model by using the AOS-DSS treatment. And so that we found the Lactobacillus family bacteria can suppress tumor growth in the mouse model. The next question is how this bacteria inhibit or suppress tumor growth. And we analyze the intra-tumor lymphocyte or the immune cells. And we found that the CD8 T cells were activated after the addition of this bacteria. And we also analyze a lot of other immune cells, such as B cells and CD4 T cells, and also innate lymphocyte. But we found that the increase or the activation of CD8 T cells are very the most important one. And we found that after the addition of this bacteria, we found CD8 T cells express high levels of granzyme B and interferon gamma, which are very important for the cytotoxic function of the CD8 T cells. And this indicates that this bacteria can activate CD8 T cells in the tumor. Then we want to know how this bacteria activated CD8 T cells. And since this two bacteria are extracellular bacteria, and we want to analyze the metabolites change related to these two kinds of bacteria, and we use the metabolomics analysis to define the metabolites related to these two bacteria. we found that after the injection of these two bacteria, we can see the downregulate of one type of the metabolites called glycophospholipids, significantly downregulated by these two bacteria. And we also analyze how this metabolites affect the function of CD8 T cells. We found that this metabolites can directly suppress the function of CD8 T cells. And then we also want to know the relationship between the bacteria and the metabolites. We found that the RG and BP, the Lactobacillus sphaeris bacteria, can directly downregulate or degrade the glycophospholipids in the tumor and also in vitro assay. And this lyso-PC can increase or promote the tumor growth. However, when we aid or inject the bacteria into the tumor, the bacteria can degrade the metabolites and also suppress the tumor growth, as well as they can increase the granzyme B and the interferon gamma production by the CD8 T cells. Summary this part, we found that the Lactobacillus sphaeris family bacteria such as Rhamnococcus glavus and Blotier, this bacteria can degrade the metabolites in the tumor environment and rescue the CD8 T cell function and promote immune surveillance and then inhibit the tumor growth. As I mentioned, that the commensal bacteria not only regulate the immune function, but also contribute to the immune development in the mucosal site. So we want to know how this bacterial coexisted or co-evolution with this immune system. And our lab majorly focused on very important lymphocyte in the mucosal surface, that is an innate lymphocyte. The innate lymphocytes are abundant in the mucosal surface. There are three types of ILC, that innate lymphocyte, and they have different types of function. ILC1 can produce interferon gamma and eliminate the intracellular bacteria, and the ILC2 mimics the function of Th2 and contribute to the Th2, the type 2 immune response. And the ILC3 contribute to the production of antibiotic peptides and also contribute to the extracellular bacterial elimination. So different types of ILCs have different functions, but they come from the same precursor. And the function of this ILCs also be regulated by the environmental signals, as also the bacteria and especially the commensal bacteria. When we use the germ-free mice, that the mice do not have any bacteria in their gut or in their body, and the germ-free mice have very little numbers of the ILCs, especially ILC1. And you may see that the germ-free, when we isolated the ILC1 in the gut of the germ-free mice, the number significant decrease in the germ-free mice. And also we analyze the epigenetic modification of these cells. So we found that the hydroxy methylation of the ILC1 are very high in these mice. When we co-host, when we take the germ-free mice out and to the SPF condition, then the hydroxy methylation of the ILC1 will be downregulated. That means that the microbiota may contribute to the methylation or hydroxy methylation of these ILC1 cells. Then we analyze also the metabolites in the gut, in the germ-free or the antibiotic treated mouse. We found that with the antibiotic treatment, there is a lot of increase of the primary bile acid, that is chloronic acid. This chromic acid is enriched in the in this germ-free mice. And when you use the chronic acid to treat this ILC1, this lymphocyte, we may say that the chronic acid inhibits the ILC1 proliferation. Also, we found that the levels of hydroxymethylation of the ILC1 is very high. When we use the chronic acid inhibitor, the can rescue the high level of the hydroxymethylation of this ILC1. That means that the microbiota can regulate the hydroxymethylation of ILC1, and also inhibit the proliferation of ILC1. Put a long story short, and we found that the gut microbiota can regulate the methylation or hydroxymethylation of ILC1, and the metabolites can inhibit the demethylation enzyme, and then contribute to the demethylation of TGF-beta receptor, and can lead to the X1 expression. And we know something about the epigenetic modification or DNA methylation of ILCs, but we want to know more about the function of methylation in the fate determining of ILC1. And we want to know how this epigenetics modification in ILCs determines their fate. Why we want to know the epigenetic modification? Because every single cell, every cell have the same genome. That means the DNA sequence are the same, but different types of cell have different functions because they have different types of epigenetic modification. So if the DNA sequence is just like a book, the epigenetic modification is like the bookmark or the content chapter of the book, and tell us how to read this book. So the epigenetic modification of the cell is very important for the fate determining of the differentiation of the different types of cells. Then the DNA methylation take part in a lot of important roles in the regulation of stem cells, such as the hematopoietic stem cells. During the differentiation, the lineage genes are opened by the loss of DNA methylation, and some stem cell genes are closed by the methylation of these genes. However, before our study, there is even no study about the methylation in the ILCs. So we want to know how the DNA methylation determines the fate of the ILC, the different types of ILC function. So the second question is, do ILCs have epigenetic imprinting during their development, and is their function determined by this epigenetic imprint? To answer these questions, we want to know the DNA methylation at the single cell level. Why? Because different types of cells have different kinds of methylation types. If you want to know the heterogeneity of these cells, you must analyze the methylation or the DNA methylation level at a single cell level. So we use the single cell multiomics and analysis of the ILC and their precursor, ILCPs. And we found that we analyzed the transcription and the chromatin accessibility and also DNA methylation level at a single cell level for different types of ILCs and ILC precursors. We found that for the ILC signature genes, such as interferon gamma or TBX21 or the GAT3 or ROS-A, they are highly expressed by the different types of ILCs, and the chromatin accessibility are also the chromatin are also opened in this region of this gene, and the DNA methylation are very low in these genes. So that means that our analysis is very similar to that what we know in LC biology. Then we analyze the heterogeneity of the ILC precursors. We found that for the transcription level, we can distinguish the ILC precursors into two subsets. That is, we call ILC1, ILCP1 and ILCP2. And the ILCP1 has higher levels of DNA methylation than ILCP2. And for the trajectory, the development pathway of ILC1, the ILCP1 and ILCP2, we may find that ILCP1 differentiation to the ILCP2. Then we use the reporter mice to analyze these two subsets of ILCP, and we found that for the ILCP, we can distinguish to the ILCP1 and P2 by the expression of UHRF1, which is a very important gene for the DNA methylation. And we also, in the vitro and in vivo experiment, we found that ILCP1 can differentiation into ILCP2 and then to different types of ILCs. Then next, we analyze the differential methylated regions of the ILCP1 and P2. We found that the DNA methylation level has very low correlation with the gene expression level. That means the lower or higher methylation level was not related to the lower or higher gene expression level. However, the hypermethylation means the low levels of methylation genes are associated with some ILC functions, such as type 1 or type 2 cell functions, which means that in the ILCP2 part, ILCP2 subset, they have their own signature, all the features of the methylation levels of the DNA. And then we next analyze the heterogeneity of the ILCP2 subset. And surprisingly, we found that ILCP2, even they are precursor cell, they have some signature, methylation signature with the ILC subset, the function ILC subset. That means that some ILCP2 have lower methylation levels in the ILC function genes and a similar signature features with ILC1, and some ILCP2 has signature features with ILC2, and also ILC3 has a similar phenomenon. That means that the ILCP2 has a heterogeneity in their methylation level, and they also have the we call it the specific methylation regions as the ILC subsets. Then we want to know this SMR, the signature methylation regions, these regions are inherent or from the precursor cell to the daughter cell. We use the methylation tracing models, and we found that if the precursor cell has the signature methylation levels as the ILC1 cells, they just restrictly differentiation to the ILC1 and similar to the ILC2 and ILC3. That means at the precursor levels, the precursors have the DNA methylation features as their daughter cells, and this methylation imprints and from the precursor cells to the daughter cells, and which indicates the DNA methylation imprinting from the precursor to the daughter cells. Then we want to know the function of DNA methylation in the precursor cells. We found if we delete this DNA methylation transferase, DNMT1, and the mice cannot develop LC subset, and the DMT1 deficiency suppresses the ILC differentiation, which means that the demethylation imprinting are very important for the ILC subset establishment and also the maintenance of the ILCP homeostasis. So to the summary, for the development of immune cells or the stem cells, there's an what into the epigenetic landscape tell us that the precursor cells, just like a ball at the top of the hill, and they roll down to the valley randomly. So the progenitor cell can generate the daughter cells by chance or randomly. But to our model, we found that the epigenetic imprints can inherit from the precursor cells to the effector cells. That means just like the precursor cell has a GPS to guide them to differentiation to the subset. So that's all my talk about how the microbiota regulates the environment of the immune cells, but also the epigenetic changes also regulate the immune cells. Thank you very much.
Science supports healthier, happier, and harmonious life.